Welcome to this video. During your science lessons, you will have already been practicing planning investigations and carrying them out. This video is going to run through some of the skills that are required to plan an effective science investigation. Throughout your science course, you are expected to carry out a number of what we call required practicals. There are 10 required practicals in the biology course. You're going to need to know the practical method for each of them, and you're also going to be learning the skills required for any scientific investigation, and that includes planning, implementing the method, presenting the results and drawing conclusions, and evaluating the evidence. The Year 9 required practicals are listed here, and you can spend a few minutes just having a quick look at which ones you're going to be doing. It may be that we do them in slightly different order, but we should get through all of these by the time we get to the end of Year 9. In Year 10, we have another set of practicals, and most of these will be carried out by the time we get to the end of Year 10, and all of these 10 practicals are recapped again during the revision course in Year 11. So to plan an investigation, we're going to start with an aim. This must be something that you can answer through your investigation and should be a neutral statement and will probably be the title of your written work. An example here might be to investigate the effect of temperature on the rate of growth of sunflowers. Now this is a neutral statement because it doesn't tell us exactly what we think we're going to find out. A non-neutral statement would be to say that investigating the effect of temperature and proving that the higher the temperature, the faster or the bigger they grow. Sometimes you might be asked for a hypothesis, and a hypothesis is something slightly different. It's a statement that will predict the outcome of this investigation. So you'll have your aim and then you have a separate hypothesis. Not all scientific investigations will have a hypothesis. If, for example, I wanted to investigate the effect of a new chemical on, for example, treating some kind of illness, I might not have any idea as to whether or not it's going to work. I might be trying a whole range of different chemicals, and in the hope that perhaps some of them might produce some interesting results. So I may not necessarily have any idea as to what's going to happen, but I've got a much more open investigation. We generally, though, have some reason for carrying out an investigation, so it's not unusual to have some expectation of an outcome. So apart from an investigation which is really just sort of doing general research to find out if there is any sort of effects, sometimes we will then pick one particular aspect and investigate it in more detail, usually because we have some belief that this might be of some interest to us. So in this instance, our hypothesis would be the warmer the environment, the taller the sunflowers. And I can then explain why this is using scientific evidence. At this point, as I mentioned previously, you might be asked to explain the hypothesis that you have given. And this is where your scientific knowledge is required. You can use theories or laws that support your hypothesis at this stage. So, for example, you could be using the theories of kinetic energy to explain why warmer objects might behave differently to cooler objects. Any preliminary studies can be quoted. So, for example, you might be using some evidence from a previous investigation that you've done, or maybe that somebody else has done. Variables. These are the factors in the investigation that can be either changed, measured, or kept the same. So here we have a whole range of variables that we could measure. A good plan needs to identify them and describe how they are being managed. So we can divide our variables into the independent and dependent. So starting with our independent, this is what you're going to change. you're going to need to decide how many tests you're going to do and answer the aim effectively. So with our independent variable, we're going to decide how many different temperatures we're going to investigate for our sunflowers. We should also state any repeats that we're going to do here. Our dependent variable is what we're going to measure. How are we going to collect our results? And what equipment are we going to require to do that? So for example, if we're going to think about the sunflower investigation, our dependent variable is going to be how tall the sunflower is going to be, so we're going to perhaps think about using a meter rule. Controlled variables, now these are incredibly important, and quite often people don't put enough information in this section. But usually in an investigation, this will make up quite a large number of the marking points. So these are the factors that must be controlled to ensure that the data is valid. And I've put that in bold here because people quite often don't understand the meaning of the word valid. If the question is telling you about designing a valid investigation, what they're really saying is that you've got to make sure that everything has been controlled. And this will then result in the investigation being a fair test. So validity is the measure of how fair the test was. You have to try to think of anything that you could have an effect on the height of the sunflowers and describe how you will control that variable to reduce that effect. So that's really important. If you don't control all the other variables, the data isn't valid and you probably haven't tested what you think. An example here would be, in the sunflower question, if we haven't controlled the amount of water that the plants have been given, this will not answer the question in a valid manner. So we could be growing our plants at different temperatures, but if we don't water them the same, then we can't be sure that it's not the amount of water that's made a difference rather than the temperature, or most likely both of them. So in this instance, some of the possible controlled variables would be the volume of water used, and note that I've used the word volume as opposed to the word amount. We want to really avoid that word wherever possible. Light intensity, so how much light we're going to shine on those flowers, so we don't want to grow some in the shade and some in the in broad sunlight. The variety or the age of the seeds used. That can make a huge difference. Some sunflowers grow particularly tall and some are designed to be quite short, so we need to make sure that we're using the same variety of seeds. The time that we plant them, we must plant them all at the same time so that they've got equal opportunity to grow. Maybe the type of soil that we're using or what we're actually growing it on. So there's a number of things that would make a difference. This brings me on to the matter of a control. A lot of people get confused between a control and the controlled variables, but they're not the same thing. A control is actually a test which is carried out to show that the independent variable is really working. And this is important because some investigations actually might be misleading, because although you feel like you're doing an investigation which is showing the effect of something on something else, in fact what really is happening is that the result would happen whether or not you change the independent variable or not. Some investigations don't require a control, and in fact you can't really work out what the control would be. So a simple one would be to investigate the distribution of some organism relative to another variable such as light, and there isn't a way of setting out an investigation to actually include a control in that. But here's an example of one where a control could be useful. If we were investigating the effects of earthworms on the speed of leaf decay, in this investigation I might set up some leaves with different varieties of earthworms or different numbers of earthworms, and then I would be measuring how much the leaves are decaying over a period of time. But what I would really need to do as well is set up a controlled investigation where I had some leaves which were set up without any earthworms at all, and that would make sure that the leaves weren't just breaking down anyway without the worms being there. If we don't set up a control, we wouldn't be sure that the leaves couldn't just be breaking down over time anyway, and that could be due to other agents of decay such as bacteria. So a control would be to set up the leaf decay experiments without any earthworms to prove that they are having an effect. Here we've got some data from the plants that we're growing at different temperatures. So we're growing them at 20, 30 and 40 degrees. And we've got 3 plants for each temperature. And if we look at the 20-degree one, we can see that there's quite a big difference in the height of these plants. So 2 of them are similar, 76 and 69 centimetres, whereas plant 2 is only 36 centimetres. So you could say that this data for the 20 degree is not very repeatable. If on the other hand we look at the 30 degrees, the data is much closer together. It ranges from 76 to 89, which although is different, isn't as big a difference as at 20 degrees. And then at 40 degrees, again, a bit of a spread of data. The data is going to be quite variable anyway, but that's always true of biological materials, and that's another reason why it's so important to do lots of repeats. So if I was doing this investigation or planning this investigation, I would probably be planning to have at least 10 plants at each temperature, and that would give me a much more representative set of data. So the data would be variable, but we'd get a lot more that was closer together. And if it was 10 completely different sets of data, then I would suggest that my data is not repeatable and I haven't controlled one of the factors well. This is a different term, reproducibility, which people often confuse with repeatability, but what it means is, can somebody else carry out your investigation and achieve very similar data? To do this, your method has to be very detailed. So to describe your investigation as being reproducible means that somebody else can follow your method exactly the way you've done it and get a similar set of results. You need to go back to your controlled variables and check that you've described how you controlled each one, and that will tell you how reproducible your experiment is. For example, have you described how much water was used to water each plant the same? So for example, if you said that every day you gave each plant 20 mils of water, then that helps to make your data reproducible. Have you named the variety of the seeds that you used? So somebody could use exactly the same seeds and get the same sort of, say, similar set of data. In order to do this, you're going to need a clearly described method. Your plan should include sufficient detail on how to carry out the procedure. Think about writing instructions to somebody in a lower school year who has never done this before. This is a very good tip because when you're describing an investigation, quite often people are far too vague in the way they put things in. So you turn a simple investigation into something that's very difficult to carry out because the information is not sufficient for somebody who hasn't done it before. So this brings me to the end of this presentation and your task. You need to now answer the questions that follow, as they will give you the practice at applying your understanding, and then you will go on to plan a valid investigation. Thank you very much for listening.